Okay. Lophotrochozoa, bilateral symmetry, and some develop lophophore, which is ciliate tentacles. It's a larval stage, and then some they have trochophore larvae. Okay. I hope the lighting is better for the group. Phylum Annelida, which this is phylum Annelida, belongs to clade Lophotrochozoa. Metamerism, repeated segments. That's what the term metamerism means. You guys mentioned that earlier. And what else? If one, the advantage of evolutionary wise, the advantage of segmentation is if one segment fails, the other ones come into play and continue. You can remove your one of, I've heard even two has been removed, of your vertebral column. When you remove it, then you still can be functional. Your fingers, you can cut one or two digits and you still have, 3 fingers and so on and so forth. Classification based on segmentation, setae. Setae is those hair-looking structures, and clitellum is used for reproduction, okay, and presence or absence of clitellum. Annuli is the ring between each metameres, which I mentioned that already a little bit in the past. So these are these grooves between each metamere is called annuli. Okay. Grooves outside is annuli. Cephalization. Some organisms they have a very well distinguished head. Earthworm doesn't. Earthworm does not have a very well distinguished head. But other organisms, you will look at them. They have some. Again, I should have put some cephalization. But if you open up the animal inside, then you will see accumulation of nerves and neurons in the anterior portion of the animal. But looking from outside, you really cannot tell which end. Ordinary people, you can. Clitellum is here, closer to the anterior portion, then that is the head. Am I making some sense? If this is this long and the clitellum is here, so I know the head is here. Am I making some sense? Closer end to the clitellum is the end, is the head of the animal. When you open it up, dissect it, I will bring it out today for you to dissect if you want to dissect earthworm, and I will be here. Please wear gloves, even though they don't have any nasty germs or anything like that. Wear gloves, and then when you're done with dissecting, put them back into trash can. Okay, the gloves, everything. And wash the utensils. I say utensils. The surgical equipment. I don't know what are the terms to use. Put them back, please. Okay, protostomium is anterior of the animal, and pygidium is the last segment of the animal. Again, on this model, you do not have it, but on this model, you do have pygidium and protostomium. Okay, pygidium is the very last segment right here. Pygidium is the very last segment, and protostomium is the very first segment. Clitellum is here, so I know the head of the animal is here. If you see a live animal, or if you're dissecting today, most all of them have clitellum. Pygidium is the terminal portion of the animal that has anus. These two are not metameres. The definition of metamere, as you saw it, it means repeated segments. So these two, the very first one and the last one, they do not have repeated segments like nephridia. They do not have the nephridia in them, so they do not refer to them as metameres. New segments are formed in front of pygidium, the very last segment, right here. Okay, so the new segments are formed from this end of the animal, opposite of what? No, opposite of tapeworms. You remember tapeworms? They have scolex, and they had the germinative zone right after the scolex, and the new segments were made. You remember that? Germinative after scolex. These are opposite. Okay. So, but again, completely two different animals. Tapeworms and earthworms. These are coelomate animals. Those were acoelomate animals. Different phylum. Okay. So where is the oldest segment and the newest segment? The oldest segment would be toward the anterior portion of the animal. That would be the oldest segment. And the newest segment is toward the posterior end of the animal, where the pygidium is, toward the posterior end. This is the anterior end toward the front of the animal. This is the posterior end toward the end of the animal. Okay. So, what else? Coelom comes from a mesoderm, schizocilus. We already talked about that at the beginning of semester. Circulatory system is closed, true heart, and open circulatory system. Might as well let me draw it here, I hope. A closed system like the one we have a heart, right? A heart has artery, and artery branches to arterial, arterial branches into capillaries, is that right? So you have a lot of capillaries, and all of these capillaries come and become venules, and venules, they come together into the heart. Vein, capillary, artery, so on and so forth. So red blood cells are always inside of a tube, right? And then eventually, when they go back to the heart, they're also inside of a tube. The heart is covered with endothelial cells. We talked about this in the past a little bit. Open circulatory system animals. Open circulatory system animals, they have heart. Okay, don't think that they don't have heart. They have artery. And what happens, arteries branch to arterioles, and from arterioles, I'm going to draw it like this, they have sinuses. They call them sinuses. And from sinuses, then blood comes, it branches off to the venules, and from venules to the vein, and back to the heart, and heart pumps. So they have sinuses, and in sinuses, the blood, red blood cells, come in contact with actual tissues, and exchange of gases and oxygen and waste material, same thing as these guys, happens in the sinuses. That's the difference. They call them open circulatory system and closed circulatory system. Which one is more evolutionary, more advanced? It's supposed to be closed circulatory system. That's what we have. We are more advanced than the closed circulatory system is more advanced than open circulatory system. Yes, sir.
Did you say they did have capillaries though? The open?
No.
No capillaries.
Open, no. They, instead of capillaries, they have sinuses.
What about venules?
They have venules, sure.
Okay.
They have venules, artery, it branches to venules. Okay, and they have venules, venules, arterioles, yes, they have all of them. They have venules, arterioles, but they do not have capillary. Lack of capillary, that's the highlight, the main difference. The rest is similarity. The rest is similarity. Okay, through circulatory system, true heart, and they all have true heart. Complete digestive system means anus and mouth, mouth and anus. Regeneration, they are the most powerful animals that they can regenerate. You cut them for your, well, we are not doing earthworm experiment, we are doing snail experiment, but if you cut them, they regenerate. Remember, if I ask you a question, somebody grab your collar in the mall and ask you a question, are these guys more complex, they have more structures, or planaria? Which one would you say? Huh? These guys, earthworm have more structures, more organs, they're more complicated than planaria. Planaria is simpler. Planaria, you learn, they do regenerate as well. These guys are the most complex animals that they can regenerate. They're most powerful as far as regeneration goes. You cut them in half, the other half will grow. Okay, peritoneum of adjacent segment needs to form the septa. Of course, you do have in these models, these are these lines in here. or septa, and when you dissect them today, if you want to, you will see these septa in here. These are Ariana. These lines are called septa. Okay, when you dissect them, you will see it. Circular muscles outside and longitudinal muscles inside, they move by means of peristalsis. Okay, review your peristalsis in this. Here is a general body plan of annelids. Generally, that's what they look like. Okay, they have septum, septa, plural, septum, singular, right here. Okay, and they have a complete digestive system, mouth and anus. This is pygidium, this is protostomium, and of course they have, in each segment, they have repeated units like nephridium. Okay, and this is the general body plan. Now, of course, Earthworm has short vestigial setae. Possibly at one time it was used for locomotion, respiration. That's what setae are. But now their function is vestigial. The term vestigial in biology means it had a function in the past, but now we don't know what's the function. Your appendix is one of them. Your appendix possibly had a function in the past, but now we don't know what's its function. It just, God forbid, if it bursts open, you have, it can kill somebody because of all the bacteria, but usually people remove it. But anyhow, there are other structures in the human body. They consider that as vestigial. It had a function in the past, but it does not have a function. Same as the setae in these guys. Not in the other organism, in other animal that I'm going to talk about here in a minute, Nerus vernus, but in the earthworm, they are supposed to be vestigial. We don't know if they have one. Here's a cross section of it. You do have a cross section slide of earthworm. Of course, the cuticle. On the cuticle, you have the epidermis. On the epidermis, you have circular muscles, longitudinal muscles, blood vessels, intestine. So everything in here, you do have them in your slides. Septum, on your slides, you might not be able to see septum. Okay, you do not see it. Visceral peritoneum, yes, you will see peritoneum in there. Dorsal mesentery, don't worry about that. Epidermis, circular muscles, longitudinal muscles, dorsal blood vessels, segment. Okay, but everything in there, I'm looking longitudinal contract. Here is explaining the peristalsis. Longitudinal muscles contract, body shortens, circular muscles makes the body elongate. Here is at the beginning of semester, Patrick asked me, I'll look at this a little bit closer, but this is usually what this diagram is explaining the excretory system, the differences, the different type of animals, they have different type of excretory system, and that is for used for higher animals, for the vertebrates, and I will, if it comes up, we'll talk about it later on, but that's pretty much like us, that's what we human are. But other group of animals, these are, none of the invertebrates that we are studying right now fits these categories. Okay, mostly for vertebrate animals. Let's first class Polychaeta. Poly means what? Many, and chaeta, it means, quiz today question. Chaeta, Bristol, is that right? That's the one I put on the quiz. Chaetae, it means Bristol. Same thing, hair, you're right. Okay, but if I put Bristol, you all know what I'm talking about. Nerus vernus, clamworm or sandworm. Okay, we do have them in the lab. The other organism, Arincula, is the lugworm, another species in this class. Largest class, mostly marine. As far as when I say largest class in this phylum, it means as far as the number of the species goes. Not as far as the size of the animal. No, not the size of the animal. As far as the different species, for this phylum, it is this class Polychaeta, different species. Okay, have well-differentiated head. You can look at the animal there, and you can tell, but from outside, you can, without dissecting it, you can tell where the head is. Okay, parapodia, paired appendages, all segments have have it except the first and last one. Parapodia are biramous. It means they're branched. That's what biramous means. And you will see some pictures, and I will talk about it. What is the function of the biramous? They do not have clitella, and they have many setae. These animals do not have clitella. Okay. Dioecious, sexes are separate. Unlike earthworm, earthworm are monoecious. That would be a nice essay question. Compare earthworm with night crawlers. Okay. Live on the rocks, burrowing to the mud and sand. Tuberculous, polychaetes, and other organisms are sedentary and spend much of the time in permanent burrows. Again, this is another organism in this class, so I gave you 3 worms in this class. Head or perostomium has eyes, tentacles, sensory palps. Sensory palps, you might see it on your diagrams or textbook or lab manual. It says palp. The function of a palp for the rest of semester, whatever, Jessica, we see it is for sensation. Okay, so palp is for sensation. It's a sensor. Sense the environment, and based on the palp, we move around, so on and so on. First segment is called peristomium. They have pigments such as hemoglobin in the blood, fertilization external, and larva is called trochophore. Quickly, gonads swell during some time of the year, and gametes go to coelom, gonadoduct, metanephridia, or ruptures from the body wall. Again, they're active at night. They call them night crawlers. They're nocturnal animals. You all know what nocturnal and diurnal means. Diurnal, like us, we human are designed to be diurnal, even though some of us are nocturnal. Right, those gang members. I better be careful what I say. You know, they might, bad boys, they might come after me. So I believe, I don't know. We human, if you read books, we human were designed to be active during day. These animals were designed to be active at night. Okay, so they call them nocturnal and diurnal type animal. Owl is a nocturnal animal, supposed to be active at night. But anyway, chitinous jaw and sensory tentacles. Each parapodia has 2 lobes. As I said, spiramous. Dorsal notopodium. I've got to show you pictures on these. And ventral neuropodium. Okay, and I will show you the dorsal notopodium. Both used for creeping, swimming, respiration. Okay, and I'll show you some pictures. Here we go. Great, finally. Okay, this is the head of the animal right here. So they have jaws. Most of what you're seeing in there, this jaw, this head portion, is retracted inside. Am I making some sense? So you might not see it like this. You can take one out of the jars and play with it and pull it out. If you want to, you can do that. But usually this head region, this very first portion of the animals, everted, it's out, everted pharynx and all of that is inside. Then you have the palp, sensory palps, right here. You might be able to see it if you put it on a dissectoscope. Eyes, again, they have eyes, but these eyes are not forming vision. We have not seen any animals, even the earthworm, have eyes in each segment. They do not have, can detect image. They detect light intensity. We talked about this in the past. Okay, then they have tentacles right here. Then these are your peripodium right here. Look at the animal. This is the anterior portion of the animal. Peripodium. What else? Okay, there is a lot of times I ask a quiz question. What is the difference between prostomium and peristomium? Prostomium is portion of the mouth part. It's not a segment of the animal. The very first segment of the animal is peristomium. Okay, so peristomium and prostomium. Cross section. You do have a slide of cross section of this animal. You do have 2 slides of peripodium. Okay, 2 slides, 1 cross section, and 1 slide of just a peripodium. Let's go over the internal structures. Eggs, you will not see that in your slides. Dorsal blood vessels, you will not, don't worry about it. But you have intestine, you have the bless your heart. You have longitudinal muscles, circular muscles underneath of epidermis, longitudinal muscles, and these are the white area right here is your coelom. It's a true coelom. These are true coelomic animals. You have true coelom. Again, this is the biramus peripodium. The top portion has notopodium. The bottom portion is neuropodium. If you look on the top portion, the notopodium, they cut it. You have a lot of capillaries. What are the function of these capillaries, you think? Very good. Respiration. So not only they use the peripodium for locomotion, also they use it for respiration. Of course, the biggest respiration portion is the top part, which is a notopodium. You have a lot of capillaries. And then a neuropodium on the bottom does not have much capillaries. It's for usually digging into the dirt and moving the animal forward. Of course, this will help also. Now, you have the setae on your slides. You have the setae. The setae are attached to a structure called aciculum. You do have it on your slides. What is the function of aciculum? It's where setaes are attached. Okay, so you have setae, aciculum, and so on and so forth. I think I went over most everything in here. Class Polychaeta, the actual earthworms. The actual earthworms. Okay, Lumbricus terrestris, night crawlers. The common name for them is night crawlers. I guess during lecture, by mistake, I called Nerus vernus night crawlers. Sorry about that. Nerus vernus is sandworms. They refer to them as sandworms. That's a common name for them. Night crawlers is the common name for the earthworms. Sorry about that. I mentioned that. Okay, Lumbricus terrestris is a scientific name. Lumbricus terrestris. Earthworms, another common name for them is earthworm. Most are terrestrial. The species, I'm not talking about the earthworm, the species in this class, most are land, freshwater, and some parasitic as well, and some marine. So you have diversity of all habitat. No head or true parapodia. They do not have the parapodia one that I mentioned in the sandworms. Okay, Nerus vernus. Anterior end has clitellum, as we mentioned that. Remember that, Jose? We talked about this.
Oh, sure.
Yeah. The anterior end has the clitellum. The posterior end is the longer. When you open it up, you see it, or you look at the dissected ones. Okay. Setae have a few. They have setae with a few exceptions. Most species have setaes. Darwin's findings, did I mention anything? No, I did not mention anything. What did Darwin say about earthworms at that time? People at his time, during his contemporaries, disagreed with him, and they thought, oh, Darwin went berserk again. I told you what berserk means, right? You guys at the front line, battlefield. Darwin went berserk again. He said that the earthworms are good for the soil. He mixed up the bottom soil with the top soil, and that is good for plants. Of course, during his time, the farmers were trying to kill the earthworm. They thought they damaged the plants, but that's not the case. Usually, what damages the plants, if it's a worm, is a nematode. It's a nematode that are damaging the plants, not the earthworm. The earthworm is good for the soil. And now there are places they actually, deliberately, they go ahead and introduce earthworm to the soil, so you mix up the soil. And of course, Aristotle, before Darwin, he said that, Aristotle was way before Darwin. He said that earthworms are the intestine of the earth. They are the intestine of the earth. That's what Aristotle said. But anyhow, each segment has 4 pairs of chitinous setae. If you want to look at it, come and count it. Okay, 4 pair, a total of 8 of cetae, exception the first and last segment. Peristaltic movement, we talked about that. Digestion, quickly, I'm going to go over these very quickly because none of this really, they're scavengers, and it means they eat everything. Sucking action of the pharynx, they have powerful pharynx. You can look at the diagram, the model here. Calciferous glands, they are, since these animals eat soil, and there is a lot of calcium in the soil, so what they have, that calcium will kill them. Remember at the beginning of semester, we talked about the elevation and lower of your calcium in your blood, just like your sugar level in your blood goes up and down, same as calcium goes up and down. In these guys, they eat a lot of soil. They take in a lot of soil. If you open them up, you will see the gut is full of soil. Okay, so they have to regulate that calcium somehow, some way. It's by those calciferous glands. And then in your models, you do have them right here next to the heart, like a leaf of some kind of plant. It's a calciferous glands. I hope you can see those. Okay, so the function of calciferous glands, it regulates calcium in the blood. Mouth in the first segment, prostomium, and anus in the last segment, pygidium. Typhlosol is the fold of the intestine. If you look here, this is the intestine. If you look here, you see a fold, the finger-like projection. We have many of them in our gut, finger-like projection, which is called what in us? It's called villi. You remember, villus is a plural, and on top of those epithelial cells, then we have another fold, which is called microvilli. Okay, so this would be equivalent to your villus, but they only have one, right? They only look at your slides. Okay, they only have one, but we have several, thousands. Villi, villus, singular villi, plural. Okay, so those that's what typhlosol is. That finger-like projection increase the surface area for more absorption of nutrient from the soil. Okay, where the soil is going to be, the soil and these animals is going to be in the brown area. You see that brown area right here? That's where the soil is going to be. And then I will talk about chorogogy cells and chorogogen or chorogogy almost look like our liver. They make glycogen, and it is outlet. If this is the intestine, the out here is one layer of chorogogen cells, and when the animal is cut, they help regenerate. They make fat, lipids, glycogen they help the regeneration of the animal as well. These fats and glycogen, which chorogogen makes around the wounds and regenerate the body. Circulatory system, as I said, is it's a double circulatory system. What I mean, when the animal moves by peristalsis, the fluid in here, in the coelomic area, right here, is moving throughout the animal. Okay? And also, they have the circulatory system, heart, they call them aortic arches. Also, circulatory system pushes the blood throughout the animal. So when I say double circulation, when the animal moves, the fluid in the coelomic area is moving, right here, outside of intestine, and also they have a complete closed circulatory system. So it's double. Dorsal runs on top, and pharynx, anus, don't worry about any of that information. Ventral receives blood from aortic arches. Again, take it to the brain and rest of body. Don't worry about any of that. Blood has colorless. Yes, worry about that piece of information. I have asked that. They have colorless amoeboid cells called corpuscles. And of course, they have hemoglobin. The red blood cells have hemoglobin. But what do you think what is the function of these colorless amoeboid cells in these animals? What do you think they are? And they're equivalent to what in us?
Kill bacteria.
Huh?
Kill bacteria.
Very good. They're part of our defense, part of the defense system of these organisms. They eat bacteria, it's like our immune system. So they have these corpuscle cells. They have these corpuscle cells to eat foreign material inside of the animal. if they are inside of the animal, in the blood system. So they're equivalent to our white blood cells, if you would, our immune system. But a lot more primitive than what we have. Remember that? You're still talking about evolution of the animals. Going up the ladder, eventually, very last day, we end up with human on the top of the ladder. They came to the party last, and they're destroying the party. These guys were in the party before us, long before us. Okay. Excretory system. Each somite have metanephridia except the first and last system, which are right here on this model, these green ones, right here. And let's see what you have to know. Nephrostome is ciliated inside of the animal. Cilia brings waste from the coelom to nephrostome and join with the waste material from blood and outside through nephridia pore. Aquatic oligochaetes released. It's something I expect you to know the difference. Maybe for the final also, but for this exam, definitely the difference between ammonia and urea, their structures. Okay, you probably learned that in your chemistry classes, but not only you have to know the difference between the structures, also I'm expecting to know the aquatic animals in general, doesn't matter vertebrates and invertebrates. Any animal that is aquatic releases ammonia as the waste product, and then the terrestrial animals on land release urea, and this is the difference. Ammonia is NH3, urea is NH2, one of the carbons inside attached to oxygen, and the carbon in the inside is attached to one ammonia on that side, one ammonia on that side. So they handle two ammonia and with the carbon inside. Anyhow, I gave you the structures. But in general, not only the structures you have to know, the chemical formula, so on and so forth. Aquatic animals, ammonia release, and terrestrial animals, urea release. Nervous system, quickly, let's go over that. Brain has neurosecretory cells, and the endocrine function and secretes neurohormones. Function is regulation of reproductions and regenerations, of course. Axon are myelinated now, so we start seeing myelination in these animals. No eyes, but have many proto-receptors on the epidermis. Each segment have proto-receptors. I refer to as eye. They are referring to photoreceptors. Same. Most defenseless organisms on planet Earth. They really, over a period of time, they did not develop any sense of defending themselves. They are more defenseless than jellyfish. Jellyfish is more simpler animal, but they have a system to defend themselves. Jellyfish, for example, the first group of animals. Even sponges. Well, sponges is not conspicuous, is not for defending themselves, it's just for support. Happen to be a type of defense as well, too. But these guys, they do not have any weapon. Nothing. The most defenseless animal on planet Earth. Okay, so that is important, again, for your final exam, and of course for this exam. Non-specialized sense organs, they have some palp, but really, and photoreceptors, that's about it. They do not have any special sense of feeling, touching the environment, so on and so on. Reproductive, monoecious. Earthworms are monoecious. The sandworms are dioecious. You remember sandworm, Nereus vernus? I talked about that. But practice cross-fertilization, of course. Female store sperm and from male in two seminal receptacles, easy to memorize. Seminal receptacles receive sperm. Seminal vesicles, they make sperm and egg. Those are the structures that make sperm and egg. Reproduction can happen during any season, unlike sandworms. Clitellum secretes mucus that holds the worms together. After copulation, a cocoon is formed around clitellum, and fertilization of eggs take place in cocoon. Young adults emerges from the egg. I'm waiting for pictures. Direct development to metamorphosis. No metamorphosis. Juvenile do not develop clitellum until they are adult. Here, quickly a few pictures. Again, I like to go, this is not in your textbook, but quickly a few things. Clitellum, anterior portion of the animal, posterior portion of the animal. Each segment has a nephridia. They release the waste material into the outside. 4 pair of setae. You remember that? 4 pairs, so the total of 8. Again, I don't think this is from your textbook either. Anterior structures, they have powerful pharynx. They have a brain. They call it a brain. Yeah, it's time to call these things brain. They are more complex. They can sense the environment. They do not have any special sense. But yes, okay, this is great. Sperm receptacles, I call them seminal receptacles, store sperm after copulation, and seminal vesicles, all of these testes in seminal vesicles, which store sperm before copulation. Make sperm and store them there. And so that's the difference between seminal receptacles and seminal vesicles. Okay, I don't believe this is crop and gizzard. This is from your textbook either, but this is not from your textbook either. Here we go. That's your textbook. The picture from your textbook. Powerful pharynx, radicular nerve. Don't worry about that. Aortic arches, equivalent to the heart. Seminal vesicles, which are seminal receptacles outside. Okay. Esophagus, of course. Crop, gizzard. They did not mention it in here, but crop and gizzard and intestine. If you're going to dissect, you should be able to observe all of these. The heart, seminal vesicles, seminal receptacles, crop, gizzard, and the rest is intestine. The rest of the animal is boring, if you would. At the beginning, you can see the powerful pharynx as well. Cross section, typhlosaur, outside layer, the yellow one is Golgi cells layer, and the inside is epithelial cells. Simple columnar epithelial cells. Typhlosaur is the fold inside. Don't worry about the blood vessels in here. Either one, dorsal. Setae, you can see the setae nephridia. Coelom, the white area is coelom. Why is it coelom? Because